So we finished talking about the animal cell in mitosis. Now we talk about a plant cell in mitosis. I can give you tips. Is that in Bio 3b, the onion root tip plant cell, it is a lot easier to identify for the mitosis stages than the animal cell. But I heard that they bought some new animal cell so that it will be also easy to identify. So for the plant cell, this is the real one. This is the cartoon. They are basically similar. You can see that, okay, this is interface. As I told you, it's a black dot, solid black dot with sharp boundary. And then in the prophase, you can see that it does have the white empty space, and the boundary disappeared because the boundary is the nuclear envelope. It already dissolved. And in metaphase, you see that all chromosomes line up in the middle. In anaphase, yes, they already separated. You can see it like this. And then in telophase, in plant cell, they do not have cleavage furrow. What do they have? They have something called a cell plate. Cell plate for the plant cell. If you see a cell plate, it's basically a straight line, but the straight line may not go through the entire cell. It may be just in the middle a little bit, or it may go through the entire middle. If you see that straight line, then this is called cell plate. Cell plate in here is actually the precursor of the cell wall. Eventually, it will become a cell wall and separate the two cells. So if you see a cell plate that may not go through the entire cell, then that is the cell. They are in the telophase. Meiosis. Now we finish mitosis. There's a reason we talk about mitosis first, because in meiosis, half of the meiosis is exactly the same as mitosis. That's why we talk about mitosis first. Now we start with meiosis. How meiosis is important? Well, we use meiosis to make egg and sperm. And the fertilization, that means the combination of egg and sperm, they form zygote. And then each gamete there is haploid, one end, and then diploid. After they fertilize, become zygote, they become two ends. The sperm contains the paternal homolog of the chromosome. The egg contains the maternal homolog of the chromosome. So by combining the maternal and paternal homolog, then you get the homologous pair. That's why it becomes 2N. Meiosis is a special type of cell division. For one reason is that it generates haploid cells from diploid parent cells. And not only that, remember in mitosis, one parent cell becomes two daughter cells, but here, one parent cell becomes four daughter cells. So the result is different. And also, the parent in mitosis, the parent and daughter cell, they are identical. But in meiosis, the number of chromosomes go from the parent diploid into the daughter cell haploid. So it is decreased by half. In meiosis, we divide meiosis into two separate, you can call it a stage. First one is meiosis 1, the second one is meiosis 2. To make our life simple, meiosis 2 is exactly the same as mitosis. So if you know mitosis previously, then meiosis 2 is exactly the same. So we want to focus on meiosis 1. The significance of meiosis 1 is there's at least three significant points about meiosis 1. The first one is this one. In meiosis 1, the chromosomes reduce from 2N to 1N. from two end to one end. This is very important. And second, and then another important is that one cell becomes two cells. So this is the meiosis 1. And at the end of meiosis, it produces 4 daughter cells, and they are all haploid. So meiosis 1, they follow very similar stages as the mitosis. You have prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis, you can see that they call it the tetrad line up. Tetrad meaning that you have 4. So in mitosis, remember, we separate the sister chromatid. In meiosis, we separate the homologous pair. So one is paternal homolog, one is maternal homolog, but each homolog, they replicate. So that you have 1, 2, 3, 4 chromatids. That's why you call it tetrad. You have 4 chromatids, you have 4 sister chromatids. That's why we call it tetrad. And what we are separating is not separating sister chromatid. We are separating the homologous pair. We are separating mother and father. So in the meiosis, I told you that there are several significant characteristics. The first one is called crossover, or we call it recombination. Crossover and recombination occurs in the prophase 1. Occurs in the prophase 1. So what happened is that, well, let me give you the analogy before I tell you the scientific explanation. Many of you have this experience when you were young. Many of you may have a bedroom right next to your parents' room, and then you heard all the stuff. One night you heard that, oh, your parents making a lot of noise. Your mom sounds very painful. But the next day, when you see them, they all look very happy. And this is what happened. Your father and your mother, they overlap with each other, and then their legs are actually touching and grinding each other. And then what are they doing? What's the purpose? The purpose of what they're doing right now is that they want to exchange body fluid, right? That's the main purpose. Similarly, when your paternal, that means your father chromosome and your mother chromosome, when they are together, what happened? Their leg also touching and grinding each other. And what's the purpose? They want to exchange genetic material in this case. So now I hope you understand why I use that analogy of your parents, because it is your parents' chromosome doing the same thing in your body. And so they exchange the genetic material so that your maternal chromosome may have some of the paternal gene, your paternal chromosome will have some of the maternal genes. Why is that? The main purpose. What is the main purpose of your parents, grinding and touching each other? The main purpose is to, I forgot, some students, they give me a lot of weird reason. Some students said that, oh, it's for fun, for sex, for whatever. It's to, the reason your parents to have sex is to reproduce, to have an offspring. In here, the purpose of touching and grinding each other of your parents' chromosome is to increase genetic diversity. This is the main reason, to increase genetic diversity. So, so remember, recombination or crossover occurs in prophase. And the main reason is to increase genetic diversity.